Hello everyone, welcome to Midterm Review Part 1. This is the same material as our first test, which is also for Lecture 1.1 to 1.4. So let's take a look at the first question. Go ahead and write down the answers as you watch this video. So question number 1 says, what are the steps of scientific method? There are all these steps. You first have to make an observation about whatever is going on around you, right? For example, right now Marshy is eating hay. That could be an observation. Then you can ask a question after you make the observation. For example, you could ask, why would Marshy want to eat hay, right? And then after you ask the question, you want to come up with some sort of idea on how to solve this problem. So that is called a hypothesis. For example, you could say, Marshy is eating hay because he is hungry, or Marshy is eating hay because it's tasty. And then you're going to conduct an experiment. In this case, it's hard to conduct an experiment with just one rabbit. But if you had a bunch of rabbits, you can do some experiment and feeding them different things. That could be an experiment. During the experiment, you would make some observations and you will collect some data. And in the end, you will come up with a conclusion. So these are the steps of the scientific method. One thing that a lot of students get wrong on the test is having the observation first. You have to make an observation before you can ask a question, and then you come up with a hypothesis. The hypothesis is not the very first step. Now, our second question is, what is an observation? An observation is anything that you can sense, right? You can see something, that's an observation. You can smell, you can hear, you can taste. Those could all be observations. When we're talking about observations, there are two types of observations. One is called quantitative observation. One is called qualitative observation. So quantitative observation is anything that you can count, and qualitative is only something you can describe without using numbers. So for example, if you were to look at this keyboard, right, a quantitative observation could be there, however number of keys on here, you can count it. There are way too many of them, right? And you could say there is one keyboard. That's a quantitative observation. A qualitative observation could be this keyboard is very long. It is gray and white and black, and it has letters on it, right? That could be a qualitative observation. And now, when we talk about an experiment, we want to make a hypothesis. But how do you make a hypothesis? You could make a hypothesis based on a question or an observation. For example, if you were to be given this question, which is the same as our old review sheet, would there be more weeds in your backyard if a lot of birds released their waste in the yard, right? So that's a question that you're asking, and then you can make a hypothesis using if-then. So the if part is always the cause, right? Cause. And then the then part is always the result, or effect. Right? So there's one driving force, which is the if, and then the result is then. So if you're looking at this question, would there be more weeds in your backyard, right? That is a result. And when you look at a lot of birds releasing their waste, that's the driving force. So if you were to make a hypothesis, you could say, if a lot of birds release their waste in the yard, then there will be more weeds in your backyard, right? And then in this sentence, there's already an if in there, which is convenient, but it doesn't always go like that. Another example that I can have is, I can ask, do carrots make rabbits happy, right? And then you can come up with a hypothesis and say, if rabbits eat carrots, then they become happy, right? See how there's the driving force, which is the carrots, and then the rabbits being happy. The happiness of the rabbit is the result. If you're simply making an observation, you can also come up with a hypothesis. For example, I have plants growing here, and you could say, if plants are grown in, well, you need to ask a question, right? You can say, why is this leaf yellow? And then you can say, a hypothesis could be, if a plant is grown indoors for 30 days, then one of its leaves will become yellow. Doesn't really matter, but you get the idea, right? Number 4. Oh, this one we already talked about, quantitative versus qualitative. One is for numbers, and one is for a description that is not a number. It could be a color, it could be a size, something that's not measurable. Next one is, what does it mean when an experiment is controlled? So when we say a controlled experiment, that means we have something to compare your experiment to. For example, If we were to do an experiment on how tall plants grow based on how much sunlight it gets, right? And your hypothesis could be if the plant is grown with more sunlight, then the plant will grow taller. So you need to set up several groups of the experiment with different amount of light with the same plant, right? But then you need certain factors that are controlled, and you need a control group as well. Those are two different things. So your control group will be the plant that's not in sunlight at all. So you can use that one in comparison to the ones that is in light. And then you also need factors that are controlled, right? So you need the same plant, same size of a pot, same type of soil, same amount of water, same amount of nutrient. And once you do that, the only factor that is changing in your experiment will be the amount of light. And in this case, because you're only testing one factor and you can see how this one factor changes the experimental result, you can say this is an experiment that is controlled because you're controlling all the other factors. Now the next one says, be able to identify independent variable, dependent variable, experimental group, and control group, right? So again, we said control group is a different thing compared to a controlled factor. So for example, over here, we have a hypothesis that says if a 3-year-old eats 1 spoonful of Nutella a day for 30 days, then he or she can't live without Nutella for the rest of the year. Obviously, this is not true, right? This is just used as an example. An independent variable, it's something that you're changing on purpose as for the experiment. You're directly manipulating it. An experimental group, just jumped. The dependent variable is what's changing as a result of your manipulation, okay? So in this case, what we're manipulating is whether we're feeding a 3-year-old spoons of Nutella or not, right? So your independent variable will be whether we're feeding the 3-year-old Nutella or not, right? Our dependent variable is the effect, what is changing as a result of your manipulation. So the dependent variable in this case will be whether or not this 3-year-old can live with Nutella. So it's always a good idea to say whether or not, because then you're making sure that this is something that varies, right? Something that can change. Another example could be, let's say we have, if a rabbit eats 5 carrots a day, then the rabbit will gain half a pound a month. All right, so for example, over here we have, if a rabbit eats 5 carrots a day, then when it's grown up, then the rabbit will gain however many pounds. Your independent variable, again, it's something that you're manipulating, right? You can't manipulate how much a rabbit gains weight directly, but you can manipulate how many carrots you feed it. So your independent variable is the amount of carrots you feed the rabbit while it's growing up. And the dependent variable will be how much weight the rabbit gains within that 1 month. Now, your experimental group versus control group is your actual experimental setup, whether you have the manipulated variable or not. So your experimental group is always something that you're testing directly, right? So for example, for this experiment, the kids that are receiving Nutella a day for 30 days will be your experimental group. And the control group is when you're removing the independent variable completely. So in this case, you could have kids who are not being fed Nutella at all. Those will be your control group. In comparison, for the bottom one, for the rabbits, if you're having rabbits eating 5 carrots a day, those will be the experimental group. And the rabbits that are not being fed carrots at all will be your control group. Okay, so I hope this makes sense. If this doesn't make sense, well, talk to someone. Go figure it out, okay? Because we're going to have a quiz in about, not for long. All right, the next one is, what are the characteristics of life? You just kind of have to remember it. Think about something that's living, right? Think about yourself. What do you have? What makes you living? You have cells, you use energy, you could grow and reproduce. You have the possibility of that. You respond to the environment, right? It gets cold, you start shivering. You maintain homeostasis, you maintain the amount of water blood sugar, balance of minerals, right? All those maintain homeostasis. Universal genetic code, you use DNA, RNA, proteins to make you who you are. And all living things use DNA pretty much. And you are, well, you can't evolve as an individual, but collectively human beings can evolve. Now the next one is, what are the units of the metric system for time? Time is second, or S. Length is meters, or M. Volume will be liters, which is L. Mass is grams, which is G. Temperature is degree Celsius. Okay, be able to convert these units. So you first need to know these are all the basic units, and then you can go from there, right? You can add prefixes such as kilo, hecto, deca, deci, centi, and milli. You can remember it as King Henry died unexpectedly drinking chocolate milk, right? Once you remember the sequence of each of these prefixes, then you can do calculations very easily. So what you want to do is that if you're doing a unit conversion, for example, if you're doing 100 meters equals to however many kilometers, right? You could be able to come up with the answers real easily. But if you're not really sure, you can always go back to the King Henry died unexpectedly drinking chocolate milk situation. So you know 100 meter is the basic unit. So that falls under the unit part, right? And then you see that kilo is 1, 2, 3 steps to the left. So what that means is you're going to place a decimal right here, and then you're going to move it 3 steps to the left as well. 1, 2, and 3. So your result should be 0.1. Okay, and you should always check your answers using this little phrase over here: small number, big unit; big number, small unit. Right? Think about if you're running a mile. 1 mile should be a lot of meters, right? Because a mile is a much bigger unit, so you should match it with the smaller number. So always double check to make sure you're right. Let's try another one. How about if you have 50 kilograms equals to however many milligrams, okay? So in this case, we have 50 kilograms. Kilo is right here, milli is right here. So how many steps do you have to jump? 1, 2, 3, 4, 5, 6, which means we put a decimal at the very end, right? Because that's where our decimal is, because it's not a point something. So in this case, we'll have a number 50, and then you add 1, 2, 3, 4, 5, 6 more zeros. It works the same way if you had a decimal, right? For example, if you have 0.3 centimeter equals to however many decameter. Okay, decameter. Okay, so in this case, the decimal is already here. You find centi, which is right here, and deca is 1, 2, 3, 3 to the left. So you use this decimal, you make it jump 1, 2, 3 times to the left as well. Let's see if this is right. 1, 2, 3. There we go. So we have the decimal starting over here, and then we make it jump 1, 2, 3 times, and we get this number right here. The next one is doing scientific notation. Scientific notation is also the jumping of decimals. So if you have a really large number and you're trying to convert it to standard notation, you simply move the decimal to the right however many times that the superscript says, right? So for example, in this case, we have 2.7 times 10 to the 5th. We have our decimal already here, right? So we're going to move the decimal 5 times to the right. So 2.7, and then we move it to the right 5 times. 1, 2, 3, 4, 5. And then we add the zeros, right? So this becomes a 0. This becomes a 0. This becomes a 0. This becomes a 0. And then we get rid of, all, this is really difficult on the computer. We get rid of all the other decimals that you don't need. This is your number that you end up with, 2.7 times 10 to the 5th. Right? We have our decimals starting over here, 1, 2, 3, 4, 5. And we have this number. If you were to have 2.7 times 10 to the negative 5th, the only difference is instead of having decimals jumping to the right, you'll have decimals jumping to the left. So we'll have negative 5th is way too big of a number. Let's do negative 3. Okay. All right, so in this case, we'll have, oh my goodness. Okay, so in this case, we'll have a number that's 2.7, and we're making this decimal jump to the left 3 times, right? 1, 2, 3, right? And then we add the zeros. We replace this one with a 0. We get rid of this one completely. What should I do? 1, 2, 3, there we go. So we made our decimal jump 3 times to the left, and what you have is 0.0027 or 0.0027, right? So this is 2.7 times 10 to the negative 3. If you were to have a really large number and you want to convert it into a scientific method, how do you do that? For example, we have 314159 number right here. What you first want to do is to find the first non-zero number and put a decimal right behind that number. So in this case, 3 is our first non-zero number. So we're going to do 3.14159, right? This is where our new decimal is, but then you have to see how many times do we need to move the decimal to the right in order to make this number the original number. So let's see. You have this decimal right here. We make it jump 1, 2, 3, 4, 5 times, which means this is 3.14 blah 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 times 10 to the 5th, right? It's 10 to the 5th instead of negative 5th because you're moving your decimal to the right. You're making this number bigger. If you're making a number smaller, then you want to move it to the left. For example, if you have 0.000000345, in this case, you first want to find the first non-zero number as well. So your first non-zero number is 3. So you're going to change this number to 3.45, and then you're going to see how many times do I have to move this decimal to this new decimal to match the old decimal, right? This is our new decimal, and then we see 1, 2, 3, 4, 5, 6. 10 to the negative 6 is our scientific notation of this really small number, okay? The next one is, what is the best unit to use for certain measurements? Obviously, you need to know these metric systems, what unit to use, but then you also want to think about a standard measurement. For example, if you're running, you could use meters or kilometers, but if you're driving, you definitely want to use kilometers, right? If you're measuring the length of this phone, you could use centimeters, things like that. The next one is, what is the difference between accurate and precision? Accurate means your data are your multiple trials of this experiment will give you data, right? If you're doing an experiment multiple times and your data are very close together, then you call that high precision. If you have data that's very close to the target, you call that high accuracy, right? If you have the data that's very close to the target and are very close together, you call that high accuracy and high precision, right? So for example, if you run 5 times and you run about 5 minutes, I don't know how running works. Let's say you run 500 meters and one is 5 minutes, and then the next time you run is 5.5 minutes. The next time you run is almost 5 minutes, right? So it's all very close to 5 minutes. You will call that high precision, but low accuracy. Let's say you're trying to run it in 4 minutes, but you're not very close to the 4 minutes, right? That would be high precision. If you're having high accuracy, low precision, it will be well, you know, if your running time is very close to 4 minutes, but the data are more spread apart, that would be called high accuracy, low precision. The next one, okay, we're almost done. The next one is, where do you need to look at when you're measuring liquid with a graduated cylinder? You want to read the bottom of the meniscus, which is a saying that you do need to know about. So at the bottom, clearly, liquid, a lot of the liquid would have either a meniscus going down or a curve of the liquid going up. With water, for example, the curve is always going down because of the surface tension and how water interact with itself and with the side of your measurement instrument. So this is cohesion and adhesion doing its thing. So over here, you want to read the bottom of the curve. That's the idea. So in this case, we have 80, right? The 80 line is right here. So the reading of how much liquid you're measuring will be about 82.7, right? If you were to say 85, that's clearly wrong. If you say 84, 1, 2, 3, 4, that's also clearly wrong. If you were saying somewhere in between 82 and 83, that would be good enough. When you are looking, when you're getting the midterm exam, it'll be a lot more clear. So for example, in this one, it's clearly 45, 40, 46, 47, 48, right? That's the bottom of the meniscus. That's the reading that you should be getting. The last part is about microscope, because the microscope has different parts to it. You have the body tube, eyepiece, nose piece, low power, medium power, high power, whatever, arm stage, coarse adjustment, fine adjustment. You should know the fine adjustment is, you should use it after you use the coarse adjustment, because that turns this, makes the stage go up and down, not as much, to help you really focus on the specimen you're looking at. There are a few questions that you should be able to answer about the microscope, how to use the microscope. So the first question says, where do you look, which lens do you start with? You always want to start with the lowest magnification, so it's easier to find the specimen that you're looking for, center it, and then you also want to focus on what you're looking at before you move on to a higher magnification. How to carry the microscope: you should put one hand on the base, one hand on the arm. What is the purpose of the cover slip? There are two purposes. One is to keep the specimen flat so that you can focus on the entire surface of the specimen. You also are using the cover slip to protect the lenses, right? The objective lenses, so that you don't get the specimen, the whatever goo that you're looking at, on the lenses. What is the reason for not using the coarse focus knob when we're on high objective lens? When you're on high objective lens, the lens itself is already really long. So if you're using the coarse adjustment, which moves the stage up and down a lot, you might squeeze the stage onto the lens and break the lens. Okay, this is it for our first ever review video for the midterm. I hope you learned something from this. I hope this was a good review for you. I hope you wrote down the notes. Go ahead and look over your notes one more time before you move on to the quiz.